And I mean, first of all, I would like to thank you, Stefan, and also Sarantis for the invitation to present our work here in this event, which I think it's a fantastic marathon of chromatin. So our lab works on oligodendrocytes, and just to introduce you all to oligodendrocytes. So oligodendrocytes is a glial cell type in the central nervous system that produces myelin, which is this lipid-rich membrane that insulates the neuronal axons and thereby allows fast transmission of the electrical impulses in the neural circuitry. Now, why we are interested in oligodendrocytes is because in diseases as multiple sclerosis, there is an autoimmune attack that specifically targets both the myelin and the oligodendrocytes. And then this leads that there is a slower transmission of the electrical impulses in the neural circuitry, leading to the neurological symptoms that the patients have. Now, we also know that a lot of the multiple sclerosis patients have in the beginning a relapsing remitting form of the disease, meaning that they have the symptoms, but then they recover. And part of this recovery is due to the fact that in the brain and the spinal cord, there is a lot of oligodendrocyte progenitor cells that I'll call OPCs that can migrate to the site of lesion, differentiate to oligodendrocytes and produce new myelin, and then leading to a recovery of the symptoms. But we also know that many of the patients in multiple sclerosis evolve to a progressive stage of the disease where the recovery does not happen. And this is partly due to the fact that these OPCs, the progenitor cells, either are absent from the lesions, or even if they are present, they fail to differentiate and myelinate. And this is one of the major questions in our lab, why does this happen? Now, in this image that I've shown you here, oligodendrocytes have one main function, which is then to produce myelin, and have been thought to be quite homogeneous traditionally. But actually, if we go back in history, around 100 years ago, when Pío del Ortega in Madrid first described oligodendrocytes, him and Wilder Penfield, they actually described oligodendrocytes as four morphological types of oligodendrocytes. And a few years ago, we had a collaboration with Stan Linnarsson's here at Karolinska Institute, where we performed single-cell RNA sequencing in the juvenile and adult central nervous system, focusing in the oligodendrocyte lineage. And one of the findings that we had was that we found 6 distinct transcriptional states of the oligodendrocyte lineage, which could then made us ask the question if this also was equivalent to functional states. And this is something that we have been investigating quite in detail, but I'm not going to talk much about this in this talk. We're focusing more on what we have been doing in multiple sclerosis and also in the epigenomic space. So following this, Ana Falcão, at the time a postdoc in our lab, that now Ana has her own lab in Portugal, and also David van Bruggen, a PhD student, they decided to perform single-cell RNA sequencing focusing on oligodendrocyte lineage in a mouse model of multiple sclerosis. And this mouse model, it's called EAE. You basically you inject the mice with a Fred's adjuvant and stimulate with a peptide, a myelin peptide. And then after a few days, the mice start developing symptoms of the disease. And what Anna and David did is that they induce this mouse model of MS in a lines where we could lineage trace the oligodendrocyte lineage. And then at the peak of the disease, they FACS sorted the oligodendrocyte lineage and did single-cell RNA sequencing by SMART-seq2. So what you can see here in this UMAP is, in this TSNA, is basically in purple the cells coming from the mouse model of MS, and in green the cells coming from control. And within the oligodendrocyte progenitor population and the mature oligodendrocytes, Anna and David could find different subpopulations of these oligodendrocyte lineage cells. Now, one of the major hallmarks of these populations was actually the expression of major histocompatibility complex genes, class 1 and class 2. And here in these violin plots, you can see here in the dotted lines, the cluster coming from control, and the solid line, the clusters coming from the oligodendrocytes from the mouse model of MS. And what you can see, there is an increased expression of this MHC class 1. But in particular, MHC class 2, there were subpopulations of OPCs and mature oligodendrocytes that express MHC class 2. Now, this was quite interesting since MHC class 1 and 2 are usually present in antigen-presenting cells, and oligodendrocyte lineage cells have not been defined as antigen-presenting cells. And this suggested then that the oligodendrocyte progenitor cells from that usually are thought to differentiate into oligodendrocytes and then produce myelin can, in certain circumstances, as this in this mouse model of MS and the inflammatory conditions, transition to an immune-like state. So we published this a few years ago, and then and in this paper we also show that at least in vitro, these immune OPCs seem to be able to activate CD4 T cells. And later on, the labs of Peter Calabresi and Alban Gauthier also shown that indeed the OPCs can transition to immune state, and in their case, they saw an interaction with CD8 T cells. So what we were quite interested in then in the lab was to see, okay, how OPCs can transition to this immune-like state. How does that happen at the epigenomic level? And for that, Mandy Meyer, a PhD student in the lab, together with Narit Saguirre, computational biologists in the lab, they decided to do, again, single-cell attack seq using, again, the same paradigm that I showed you before. So a mouse model of MS, they induced in a line where we could lineage trace the oligodendrocyte lineage, and then we could, by fax, isolate the populations, and then perform attack seq. Now, Mandy and Enrichment used three different platforms, the attack seq. They used originally a plate-based platform that was developed by Howard Chang's lab. They also used the 10x genomic single-cell attack, and more recently they've used the 10x genomic multiome attack RNA platform. And this is the single-cell attack. This is how the data looks like. So again, we have in this UMAP the progenitor populations where we could differentiate the control and the mouse model of MS OPCs, and the mature oligodendrocytes, and we also actually had like some microglia in our populations. And this is how the data looks in the genome browser. So this is an MHC class 1 locus, and here every single row is a cell. And what you can see here is in microglia, you have chromatin accessibility at the promoter regions of these MHC class 1 genes. Well, for instance, in OPC control and the oligodendrocyte progenitors control and mature oligodendrocyte control also, you don't see much accessibility, but in the mouse model of MS, then you see an increase in accessibility in the regulatory regions of these genes. But what was also quite interesting was genes like this one, TAP2, where actually we see a chromatin accessibility across the board at the promoter, suggesting that even in control oligodendrocytes and progenitors, some of these immune genes might already have the chromatin open. So Mandy and Enrichment then also perform then the single-cell multiome, and in the same paradigm. And here again, it's UMAPs showing how the RNA and the attack, the different populations of mature oligodendrocytes and OPCs from control and DAE. And here how it looks in the genome browser. Again, the same locus as I showed you before, MHC class 1. And as we had seen before, we can see chromatin accessibility, for instance, not so much in this loci in control mature oligodendrocytes, but then there is an increase in the mouse model of MS. And at the RNA level, we also can see that these genes are mainly expressed in the mouse model of MS and not so much in control. However, then we also saw like some other classes of immune genes that exhibited, like I mentioned before, this kind of chromatin priming, that there is a chromatin accessibility even in control populations, oligodendrocyte progenitors and mature oligodendrocytes, even if there is not so much expression. So we then went to investigate if this chromatin priming was a more widespread phenomenon in the immune genes. And since we had like single-cell RNA sequencing data and single-cell attack-seq data, then we could actually, in each of the populations, OPCs and mature oligodendrocytes, we could then separate by categories, by types for instance, genes that had like increased RNA expression and increased chromatin accessibility, and this is type 1 genes. And what was interesting to see is that within these type 1 genes, we could find already some genes that already had the prime chromatin accessibility, but still had an increased chromatin accessibility in the mouse model of MS. But then we also have like genes like the type 2 that also have an increase in RNA expression in the mouse model of MS, but the chromatin accessibility does not change. And both in the OPCs and the mature oligodendrocytes, what we've seen is that many of the genes that are under this type 1b and type 2a categories are actually genes that are involved in antigen presentation, interferon alpha and beta response, and so forth, indicating that indeed there is a cohort of genes, immune genes, that are already chromatin primed in mature oligodendrocytes and OPCs in control. In our previous study, we actually had seen that if we treat primary OPCs with interferon gamma, you have the induction of this MHC class 2 genes. So then what Enrichment and Mandy did was to treat primary OPCs with interferon gamma and then check by bulk RNA sequencing and bulk ATAC-seq what was their profile. And what they observed is again the similar what we had observed in our single cell ATAC-seq data in the mouse model of MS, that there was category type 1b and type 2a genes that were basically genes that had already, for instance, type 2a that did not change the chromatin accessibility but had an increase in RNA, or genes that have an increase in chromatin accessibility but they were already like an intermediate priming stage at control conditions. So our data suggests that OPs, the progenitor cells and the mature oligodendrocytes have already some chromatin priming at these immune genes, but that some of them are only, that does not translate immediately in transcription. So suggesting that there is alternative mechanisms by which there is an induction of this transcription. So then what Mandy and Enrichment did was then investigate, for instance, CTCF binding, H3K27 acetylation, and also they did high chip and used the ABC model to see how were the promoter enhancer interactions in OPCs in control or OPCs treated with interferon gamma. And what they observed was that there was an increase in binding of, for instance, in this MHC class 1 and MHC class 2 loci, there was an increase in binding of CTCF, an increase in the H3K27 acetylation signaling, of chromatin accessibility, as we had seen before, but also of the promoter enhancer interactions. They also then performed cut and run with H3K4 3 methylation and H3K27 3 methylation. And again, in this MHC class 1 and 2 gene locus, what they could see that in control, you already had like in some of the genes H3K4 3 methylation, but also in some of the genes you also have H3K27 3 methylation, which could suggest a bivalent state. But then when treating with interferon gamma, what they could see, for instance, in some loci, was a reduction of the H3K27 3 methylation. We could suggest a role in polycomb in repressing the expression of immune genes. So what then Mandy and Enrichment did was then to treat primary OPCs with an EZH2 inhibitor in the presence of interferon gamma, and then perform bulk RNA sequencing. And what they observed was that while EZH2 inhibition by itself led to changes in genes involved in central nervous system differentiation, for instance, with interferon gamma and EZH2 at the same time, they would see like genes involved in antigen presentation. And here is an example, again, in the genome browser of some of the immune genes that seem to have increased expression upon EZH2 inhibition in the context of interferon gamma, indicating that at least H3K27 3 methylation deposition might be involved in the repression of these immune genes in OPCs. So we also were interested to investigate what kind of transcription factors might actually be modulating this expression of immune genes. And for that, we applied ChromVar in order to be able to see transcription factor motif accessibility, even if there were changes in the different populations that we were investigating. So again, here in each row, you have a transcription factor motif, and in each column, you have the cells from the different populations that we have identified. And what you can see, for instance, here was that there was an increased motif accessibility to transcription factors of the SOX family in mature oligodendrocytes and OPCs, which is as we would expect, since these are transcription factors that are important for the identity of oligodendrocytes. Also, we have, like, for instance, for microglia, genes of the HETS family. But then what was quite interesting was, for instance, here in the mature oligodendrocytes and OPCs coming only from the mouse model of MS, that we had several transcription factor motifs from, for instance, from the BZIP family. And here we have some examples, for instance, of, again, an overlay of the transcription factor motif accessibility. And you can hear here on the UMAP that I've shown you before. And you can see here that here in the mature oligodendrocytes in EAE, you have an increased accessibility of STAT1, and in some populations also of the BACH1 transcription factors, which are transcription factors that have been involved in immune regulation. So what then Mandy and Enrichment decided to do was then to treat primary OPCs with knockdown both STAT1 and BACH1 in primary OPCs in the context of interferon gamma treatment. And what they observed was that, again, with treating in the interferon gamma, some of these immune genes like CD274, which is PDL1, and NLRS-C5, they were increased by interferon gamma treatment, but upon siRNA with STAT1, there was a reduction of their expression, indicating that STAT1 might be relevant for the interferon gamma induced increase. They also then, Mandy then did cut-and-tag against interferon gamma, against the STAT1 in OPCs treated or not with interferon gamma, and what she could see in this loci was actually an increase in binding of STAT1. And actually, when then Enrichment and Mandy went to investigate the genes that were up or down regulated upon STAT1 siRNA, they could see that in these genes there was actually, upon treatment of interferon gamma, there was binding of STAT1. This was with STAT1, but they investigated BACH1, and with BACH1, actually the picture was different because, for instance, for this subset of genes like CD74, there was an increase upon treatment with interferon gamma of the expression of this gene in OPCs. But with BACH1 knockdown, what we saw was even a further increase, suggesting that BACH1 seems to have in this context opposite role that we have been seeing here for STAT1. Now, this data that I've shown you so far is data that we have obtained in the mouse model of MS and upon treatment of mouse OPCs with interferon gamma. So then what we also were quite interested to investigate is the relevance of this for human disease and for MS. So here is again the MHC class 1 locus that I've shown you before with chromatin accessibility in several of the promoters, but only expression in the mouse model of MS. And so what we decided then to do was also do single cell multiome in the human brain gray matter. And so what we can see in these UMAPs is again the cell types from the RNA and the attack, and we could recover the major cell types in the central nervous system. But then we look at the same locus that we had looked before, and what we can see is again, as we had seen in the mouse model of MS, but also in control OPCs, that was in oligodendrocytes and OPCs and microglia, there was chromatin accessibility at these promoter regions. But what was even more interesting is that actually even neurons and astrocytes presented chromatin accessibility, suggesting that this chromatin priming of immune genes might be more widespread than previously thought. This is not in all the immune genes. I mean, we have immune genes that, for instance, we don't observe chromatin priming in oligodendrocytes and OPCs, but we do in other cell types. And in this particular case, we then went to see in the same locus in our mouse data, mouse EAE data, and what we could observe was actually that there was an induction of chromatin accessibility in the mouse model of MS compared to control, suggesting that in inflammatory environment there might be an induction then of the chromatin accessibility of this loci, of this locus. And then actually, when we go back to our primary OPCs data where we treated an interferon gamma, we could indeed see that there is an increase upon interferon gamma treatment, increased chromatin accessibility and also expression of this gene, suggesting that there is the priming, but also the context of the disease might also actually contribute to the increased accessibility at these immune genes. Now, this gene that I mentioned was particularly interesting because there is actually a MS susceptibility SNP located in this location. And a few years ago, the International Multiple Sclerosis Genetic Consortium, they published a new GWAS study with a list of variants that were associated with MS susceptibility. And in this study, they also compared with bulk RNA sequencing data and could define that apart from the expression of genes associated with these variants in immune cells, also microglia in the brain also expressed, could be associated with these SNPs. Now, the fact that we were seeing chromatin accessibility in immune genes in the oligodendrocyte lineage suggested that perhaps oligodendrocytes might actually also have a role in this susceptibility. And then what Enrichment did was then she went to investigate the overlap between the genomic regulatory elements that we found in our data sets and also these GWAS variants. And what she could find was that indeed she could find microglia as one of the associations, as had been previously described, but actually she could also observe astrocytes and, for instance, oligodendrocytes being with the overlap both in the multi-omic attack data, but also in our mouse single-cell data sets. And this is an example of several SNPs that have been associated with MS, and some of the overlaps that we have been observing in our data sets. And to give you a further example of this, this was not only at SNPs that were located in coding regions, but also SNPs that were located, for instance, in intronic regions and intergenic regions. And this is an example of a SNP that was quite interesting for us. And this is a gene CYP24A1 that is coding a protein that is involved in vitamin D metabolism and has been associated a SNP in this loci, in this locus, to multiple sclerosis susceptibility by vitamin D metabolism in neurons. So what we found was actually that there is another SNP here in this locus that actually does not have so much chromatin accessibility, but then when we look at the same locus in our mouse data, actually it overlaps in our OPCs with the locus where they have a lot of H3K27.3 methylation. And then what we observed was that upon interferon gamma treatment with our high chip data is that there is an increased interaction of this locus with a neighboring locus, which is in the promoter of the BCAS1 gene. And this was quite interesting because BCAS1 is actually a gene that is involved in the early oligodendrocyte differentiation. So our hypothesis here is that that perhaps these SNPs that are located at this locus might not only affect this gene, but actually might also be affecting this neighboring gene in oligodendrocytes. So now what we are, we want now to correlate a lot of this epigenomic single-cell attack-seq data that we have obtained with a single-cell RNA sequencing data to make further inferences from our data. And this is something that we are doing actively. When a few years ago, we did a first study where we did single nuclear transcriptomics of 20 postmortem samples from controls and individuals with progressive MS. And this was done in collaboration with Anna Williams and Charles French Constant in Edinburgh, and also Diraj Malhotra at Roche. And now we are actually doing a further study with a much larger cohort, where in order to investigate both in the gray matter and in the white matter, the expression profile of the different populations. And then we will be able to correlate with some of this epigenomic data that we have been observing this possible connection at SNPs present in the oligodendrocyte lineage. But one thing that we are also investigating is actually the transcriptome during oligodendrocyte development, that we think that also might have an important role here. And just to backtrack a little bit, oligodendrocytes, they are generated during development from neurostem cells, and then they proliferate throughout embryogenesis in the mouse, and only start differentiating at postnatal stages into oligodendrocytes that give rise to myelin and the myelinate axons. During the mouse development, this transition between neurostem cells and embryonic OPCs actually has been shown to start occurring very early on at embryonic day 12.5 in ventral regions of the brain in the mouse. And then there is like waves of specification from ventral to dorsal, finishing at embryonic day 17.5. And the previous studies from our lab that Sueli, David, and Daria have done using single-cell RNA sequencing actually show that these different waves of generation of oligodendrocytes, they don't originate OPCs directly, but actually they originate a pre-OPC stage in between the neurostem cell and the OPC that then converges into the OPCs at postnatal stages. Now, when we go to human, there has most of the literature has been showing that actually oligodendrogenesis seems to occur quite late. And in this paper from Arnold Quickstein from last year, they also observed these pre-OPCs that we had observed in our mouse studies. But again, this was observed in the second trimester of human development. And what David in our lab went to investigate was actually whether there was oligodendrocyte lineage cells, OPCs, that were derived actually in early stages of human development, for instance, in the first trimester. And indeed, she could observe that in these early stages, that they were like a pre-OPC population that was giving rise to these SOX10 positive oligodendrocyte precursor cells. And David went then using this together with Mats Nilsson lab, using the technique of spatial in situ sequencing that could target several genes, and could identify indeed that there was a ventral population early on during development that can give rise to these pre-OPCs in human development. And again, coming back to epigenomics, then Fabio Paul in our lab then also went to these early stages and did single-cell attack seq. And what he could again identify was pre-OPCs and OPCs that were, for instance, had chromatin accessibility in the SOX10 locus, indicating that indeed at the epigenomic level, there is a population of the oligodendrocyte lineage cells that it's specified very early on. And this is some of the data that, again, that we want them to correlate with all our epigenomic data, also in the context of our studies in MS, in order to be able to see if there is a correlation between the chromatin accessibility. Now, most of the studies that I've shown you so far are then basically in single-cell attack seq for epigenomics. But we also are very interested then on the histone modifications and to characterize them genome-wide. And as many of you know, one of the standard methods for profiling histone modifications is ChIP-seq, based on an antibody recognizing your histone modification of interest, performing immunoprecipitation, and then sequence the DNA that is associated with the antibody. But at least in our hands, and ChIP-seq always had like some problems of background, but also requiring a large number of cells. So a few years ago, the lab of Steve Henikoff developed cut-and-run, which does not require immunoprecipitation, but actually requires the presence of this fusion protein, protein A MNase, that will bind to your antibody of interest of the organizing your histone modification, and then will cut the DNA, and then one is able then to do sequence your library and examine which chromatin, which DNA is associated with your histone modification. Steve's lab also developed later on cut-and-tag, in which instead of the MNase, he uses a TN5 transposase, which is very much transposase that is being used in all the ATAC-6 studies that I've shown you before. And in this case, is not only the TN5 would cut the DNA, but it also insert directly the Illumina adapters that would then allow the sequencing in a much cleaner way. And then Steve at this point already showed that at least at plate base, it would be possible to do this technique at the single cell level. So Marek Portozovic in my lab was quite interesting in this kind of technology. And since we had previously, we had a lot of experience with TN5 transposase and also with the 10x single cell attack protocol, Marek decided to try to see if it would be possible to optimize the cut-and-tag protocol and couple it with the 10x chromium single cell attack. After several optimizations, Marek was able to establish that it was indeed possible with cell lines, for instance, to do this and to identify the different types of different identities of the cell lines using single cell cut-and-tag. And then what he did was to implement this technology in a complex tissue, in this case, our tissue of interest, which is juvenile mouse brain, in a line that we could again lineage trace the oligodendrocyte lineage and glial cells. And again, this is how the data looks like at the genome browser. You can see here, again, each row is a single cell. And here is a SOX10 locus, a transcription factor, very important for the oligodendrocyte lineage. And what you can see is that it's several cells that have a lot of H3K43 methylation at this locus, but others don't have. When Marek used H3K27 methylation, he observed again the same, that some of the cells had a lot of the signal at the near promoter regions, but some others didn't. And then what Marek did was went on to cluster the data based on the deposition of these histone marks genome-wide. And with this, Marek was able to actually to identify the major cell types present in the brain based on H3K43 methylation, H3K27 methylation, and then Marek also extended to H3K27 acetylation and H3K36 methylation. And again, he could identify the major glial cell types, for instance, with these ones. And then he also extended this to transcription factors, some of which did not work and some of which, like OLIG2, work, and then we could separate low binders from high binders, being the high binders, the cells of the oligodendrocyte lineage, and also RAD21, which could also separate the different subpopulations of glial cells. Now then what Marek did, and I've told you before in the beginning of the presentation, that at the single cell RNA sequencing level, we could distinguish 6 different transcriptional distinct states of the mature oligodendrocytes. But actually, when you look at the single cell cut&tag data, we could not identify these different states. We could just basically see OPCs and mature oligodendrocytes. And this, I think, we think one of the reasons of this is the depth of the data, that it's also very sparse data. But actually, when Marek went to use a metagen approach on the integrated data, he could actually see if he would take metagens for OPCs, that they would have increased H3K43 methylation signal in the same region where the OPCs are present, and so forth, also for the mature populations of oligodendrocytes, indicating that, yes, indeed, the epigenomic information is there at H3K43 methylation of these substates, but at this point, we still need the assistance of the RNA sequencing data to establish these substates. One thing that we also noticed was actually that a few years ago, the lab of Enbrunet had described that the H3K43 methylation breadth was linked with cell identity. And indeed, in our data, we could see that marker genes from the different populations had a higher breadth. So what Marek did was then do the pseudo-time of the oligodendrocyte lineage from OPCs to mature oligodendrocytes. And then he actually ordered these cells and see in the mature oligodendrocyte promoters how was the breadth. And what he could see was indeed from OPCs into the mature oligodendrocytes, he could see this increase in the breadth of H3K43 methylations, as had been suggested in Anne-Brune's paper. We also then, Marek and Mukund in the lab also used the data to try to predict promoter enhancer interactions using the ABC model that had been described before, and also using the high chip to validate these predictions. And we got, for instance, from these predictions around 200,000 loops. And when we compared with high chip data from oligodendrocytes versus MS embryonic stem cells, we can see that there is much higher pileup in the loops that we have identified with the oligodendrocyte loops. And then what we can actually do is filter the data basically on the presence of H3K43 methylation at promoters, or the presence of RA21, and actually from these 200,000 loops, you get 5,000 loops. And when we overlay our predictions with high chip data, we can actually see here in black that there is quite a good correlation between our predictions and what we see in the high chip data. Okay, now just to finalize, so we've been focusing a lot in this single cell cut-and-tag technology, but one thing that we are, of course, it's also quite interesting is also actually do it at the spatial level. And I think, and in order to do this, we actually started the collaboration with Rongfan's lab, Yanxiang Deng at Yale University. And Rong and Yanxiang had a few years ago this paper where they described DBit-seq, which is a novel technology that would allow spatial resolution in the RNA sequencing data. And actually, Rong and Yanxiang developed now a new technology where they have adapted this technology, but you utilize cut-and-tag. And here you have a brain from P21, and what we observe in this data set, and we did H3K43 methylation and H3K27 methylation, and we could identify different clusters that were specifically positioned in different areas here in this brain section. And then when we went to look at the deposition of some of the marks, we could see, for instance, here in this area here, there was a lot of deposition of SOX10, a marker from oligodendrocytes. And this structure corresponds to the corpus callosum, where a lot of oligodendrocytes are deposited. For instance, for H3K27 methylation, we saw in the same area a lack of H3K27 methylation or at the promoter of ATPR2, which is another marker of the oligodendrocyte lineage. What then Yanxiang and Marek in my lab did then was also then to integrate the single cell cut-and-tag data that we had generated with the spatial cut-and-tag, and we could indeed confirm some of these identities that were suggested by this spatial pattern. And just to give an example of this, I mean, for instance, some interesting information that we obtain. For instance, here, CUX2 is a marker of one of the layers of the superficial layers here in the cortex. And what we can observe here is that there is an absence of H3K27 methylation here in this layer at the CUX2 gene. And for instance, this PCL11B is a marker of some more interior layers like layer 4 and 6. And again, we can see the same pattern, which suggests that then polycomb might be involved in the maintenance of these layers or at this P21 stage. Okay, now I'll finalize my talk. So we've been using a lot of single cell omics technologies in our lab, and these have been allowing us to identify new aspects of biology of oligodendrocytes, both in development, but also in multiple sclerosis. And one example of this is this transition for an immune state. I just wanted to point out that we have in our website all our data sets where you can explore the chromatin accessibility or the expression of your gene of interest in these shiny apps in the different populations that we have shown identified. And then to finalize, I would just like to thank my lab that has performed this work, particularly Marek for the SCUT&Tag, Mandy, and then edits for the MS model and single cell attack, David and Fabio for the human filter data, and also Mukund also for the collaborating with single cell cut-and-tag, and also our collaborators that have been instrumental here in this work, and all our funding agencies. And with that, I'll finalize, and I'll be happy to answer any questions that you might have.
Thank you, Goncalo, for your talk. Really great talk. And yeah, please put your questions into the question and answer box. Sarantis, do you already have a question?
Can I start, Goncalo, with that? It's amazing. Congratulations, great, really amazing data and amazing protocols. I am really intrigued from the spatial cut-and-tag protocol.
Yes.
Would you like a little bit to give details about technically how you will do it? I mean, it's microfluidic based.
Yes.
And as far as I understand, you are able to identify in a single gene level the pattern of histone profiling in different areas of the brain, for example, right?
Yes.
But how can you technically do this actually?
So this is based in ligation. So basically, what Ron's lab has developed is a method where you can actually put ligate barcodes in the specific region that you are identifying. I mean, the resolution that we have so far, I mean, for the single cell RNA sequencing, he has a resolution of 10 micrometers. Now he has a resolution of 20 micrometers for the single cell cut-and-tag. So basically, and with that, you are able to identify the region, basically where the specific region that your signal comes from, and then you can map back and basically see how your cut-and-tag signal. So, and I mean, Ron also has observed, I mean, in some of the regions you can see individual cells, some of the regions you won't have because 20 micrometers, it's still, so some regions you will have and some will not. And then that's why you can see in our data that some of them, some regions we can clearly identify the populations, and some of them it might become one or two cells. Then it becomes a little bit more difficult to identify.
Great, thank you very much. Thank you.
There is one more question, and then we need to move on to Efrat's talk. It seems like some genes have high expression but low chromatin accessibility. How do you understand the underlying mechanism for this? And how strongly do you see correlations between gene activity and expression from individual single-cell ATAC-seq and single-cell RNA-seq, probably?
Yes. This is a puzzling question that we also have, that we do when we were dividing in these subtypes. So we do see like a few subset of genes that we do not see so much chromatin accessibility, but we do have an increase in gene expression. While the other ones that have a chromatin accessibility, it's easier to explain. Those ones, we don't really have a good explanation of how is this happening, how you can have low chromatin accessibility and still an increase in gene expression. It could be a technical aspect of the data, but I'm not sure if that's the reason why. That's a very good question, very well spotted.
Okay, perfect. Then Goncalo, I will